The formation of the urinary system. The nephrogenic cord is a component of the urogenital ridge that gives rise to the urinary system. In this cross-sectional view of a 20-day-old embryo, we can identify the developing neural tube and three distinct areas of mesoderm, namely the lateral, intermediate, and paraxial mesoderm, that extend bilaterally along the axis of the embryo. The nephrogenic cord originates from the intermediate mesoderm. During embryo folding, the mesodermal components undergo repositioning, with the intermediate mesoderm coming to rest dorsal to the early longitudinal body cavities. The earliest indication of primitive nephrons in the primordial kidney can be found in the nephrogenic cord by the 4th week. This bilateral pronephros can be found at the most cranial portion of the nephrogenic cords. A pronephric duct also forms adjacent to the pronephric tubules. While these early tubules disintegrate, the duct remains and contributes to the formation of the next stage of development. Let us take a closer look at the development of the intermediate stage of urinary system development. We can identify paired mesonephroid, the intermediate excretory organs, and the developing mesonephric duct. We can also identify the vitelline duct, the allantois, and the hindgut, all of which were formed during embryo folding and concomitant inclusion into the embryo of a portion of the umbilical vesicle or yolk sac. Over time, the mesonephric duct continues its development, reaching and connecting to the distal portion of the cloaca, an expanded portion of the enveloped umbilical vesicle. The mesonephros develops a small number of primitive nephrons. The definitive kidney, or metanephros, develops from a bud of the inferior mesonephric duct. Interactions between epithelial and mesodermal tissues drive the formation of the kidney through a complex budding process. This results in the formation of many collecting connections, as well as growth of the associated functional components. The interaction of the epithelial tissue, identified here as the metanephric diverticulum or ureteric bud, and the associated mesodermal or metanephric mesenchyme, leads to development of the nephrons. Development of the nephrons continues until approximately week 36, with the number of nephrons reaching between 200,000 and 2 million. Although no additional nephrons are formed after this time, functional maturation continues after birth. As the embryo's body elongates, the kidneys change position, beginning in the pelvis and eventually coming to rest inferior to the suprarenal glands. The portioning of the cloaca is critical to the development and separation of the developing embryo's genital, urinary, and gastrointestinal systems. The formation and extension of the urorectal septum toward the cloacal membrane provides this separation of the urinary bladder and the hindgut. The urogenital sinus, which contributes to the formation of the bladder, is divided into 3 parts: vesical, pelvic, and phallic. Further development of the urogenital sinus into vesical, pelvic, and phallic parts begins to provide the primordium of the urethra. In males, the mesonephric duct becomes the ductus deferens, with connection to the developing testes and initially to the bladder. Over time, and with growth of the bladder and other tissues, the position of the ductus changes and then contributes to the formation of the ejaculatory duct. In males, the urethra is formed from the urogenital sinus and ectodermal cells of the glans penis. In females, the urethra is formed entirely from the urogenital sinus.